So first of all, I would like to thank organizers. And so I will be talking about how interplay between cohesion kinetics and polymer relaxation modulates both chromatin structure and dynamics. So I will be mostly focusing on this TAD level organization, where this loop extrusion plays a very crucial role. So as you know, this cohesin are ring-like proteins, protein complexes that can bind on chromatin, and they extrude loop until they encounter these barriers like CTCF to form these loops. So many recent live cell imaging experiments have shown that uh, these structures are very dynamic. With many of these are in partially extruded state, and also the live cell imaging also shows that this chromatin loci can reorganize very fast compared to the standard polymer models, which are used like fractal globule and ideal polymer. So that's why loop extrusion can play some role in reorganization of chromatin polymer. So these are the main questions that we want to answer. So can a model consistently explain both static 3D organization as well as the dynamic properties? Also, how the relative time scales of this cohesion kinetics and the polymer relaxation affect the chromatin structure? And how do they also affect the dynamical scaling exponents that we study in polymer physics? So we have this simple minimal model where uh, we have the weak attractive interactions between these chromatin beads. So that also we can tune the attractive strength. We have the connectivity along the polymer backbone, which is the spring energy, and also a spring between the cohesion extruders that forms the loop. And we have the kinetic events of this binding and dissociation, where cohesin can bind and dissociate with some rate, and that will decide the residence time of cohesin on the chromatin and also the cohesin density, while the sliding rate decides how fast the cohesins form this loop. So first we compare with the already existing experimental data to validate the model, where we can recover the same TAD-like structures when we have both CTCF and cohesin. When we remove the CTCF boundary, the TAD structure goes away, but contact map is still dark, and it has a lower 3D distance. And when we remove the cohesin, the 3D distance increases, and also you can see that contact map becomes lighter. And we also recover the boundary probabilities. So uh, now many models already in the literature talk about the processivity, where processivity is here in our model, it is the ratio of the sliding rate to the binding and dissociation rate, and it will decide the average loop size. So if you have low processivity, the average loop size will be small, but if you have higher processivity, the loop size will be big. So many studies propose that processivity will determine the chromatin structure. So for example, here I am showing contact maps for a fixed processivity, but different residence time of the cohesin. So if you have low residence time, so in all of these cases, the contact maps look very similar. So one would assume that the chromatin structure is also same. But if we consider the low residence time, we see that those loops are very compact in comparison to, as we increase the residence time, those loops are opened up. So we think that polymer relaxation has some role to play. And you can see that also in this movie, where the extruders will bind and dissociate much faster on the left side because they have a slower residence time. So the polymer will not relax in that time, and structure is compact. On the other hand, in this case, the extrusion activity is very slow, and polymer relaxes and takes this open structure, coiled structure. So to quantify this, we calculate the relaxation time. So we take this position vector of these beads, which are separated by some contour separation, and we compute how the correlation of this vector decays with time to get some characteristic time of the relaxation time. And then we can plot it as a function of genomic separation. So this is without extrusion. So here for on average given loop size, we can have some relaxation time, which is of the order of 10 power 6 in simulation units. And once we have the extrusion activity, we see that the relaxation time decreases, which means that chromatin relaxes faster, so it can reorganize faster, and also the scaling exponents also decrease. So, which can also explain why these, which is observed experimentally, the lower scaling exponents in the experiments. Maybe loop extrusion can play some role. And now we can compare this residence time with the relaxation time. So if the residence time is comparable to the relaxation time, we'll have a higher RG and structures will be like coiled structures and open. While if we have a residence time much smaller than the relaxation time, then the RG is less and structures are compact. So this is again consistent for different processivity values. So for a fixed processivity, we always see that when we have a lower residence time, the structures are compact. Apart from that, we also measure the dynamical exponents like single locus MSD. So we see that we vary different parameters in our model, and we observe that the scaling exponents vary around 0.5 with minimal change. On the other hand, the two locus MSD also shows some change. So it has two phases. So initially, at smaller time scales, the two loci will diffuse independently. But at longer time scales, due to these correlations, it will saturate to some average value. And again, the transition will happen around the relaxation time that we computed earlier. So all these plots again suggest that the extrusion activity overall reduces the scaling exponents and helps the chromatin to organize faster. So in summary, our model can capture both static and dynamic properties of the chromatin polymer. And the interplay between this polymer relaxation and the cohesin kinetics can modulate the chromatin structure. And we also measure the dynamical scaling exponents and compare with the experimental data. So this is again available on the bioRxiv. And I would like to acknowledge Professor Ranjit and also the collaborators, Professor Sunil and Saudamini, and also lab members. So I can take questions.
Thank you. Thank you for an interesting talk. Questions? No questions?
Yeah, I was just curious. So this impact of the residence time and so on, does that depend on the interaction energy that you put in? So if you put in higher interaction energies, does it play less of a role?
So with interaction energies, we have fixed interaction energy in this case. So we haven't varied much, but I would assume that once you increase the attractive interactions, RG overall will decrease, so the difference will be much less.
So in this model, following Mithun's question, let's say you have this long loop that has been extruded. If cohesin has multiple stalls in between, would that be faster in this case? I mean, instead of extruding at one speed, let's say you had enhancer promoter in between that also have some attraction for cohesin. Would that matter in these models?
So right now, our model, we have just uniform speed. So on an average, it will have some speed. So we haven't placed any roadblocks like that. But one can incorporate that and study how it will affect the dynamics.
Thank you.
Mine is actually, I don't know whether I missed it. Did you consider different extrusion rates also?
Yeah.
Okay. And that has an effect?
Yeah, so processivity, so for example, here you can see we vary the sliding rate.
Time, no?
This is sliding rate. So processivity will be effect of both sliding rate and dissociation rate. So we can vary both and check how it varies.
Okay. Any other questions? If not, then let's thank the speaker. Thank you, Sangram.